Hey guys, welcome. Welcome. This is our second lecture session. Remember, this was designed based on your needs, and we're going to divide this up into 4 sessions. This is the second of the 4 sessions. And remember, you could join me to review. We'll have 2 review sessions twice a week. So this coming Tuesday and Wednesday, we will have a review session of the lecture that we're doing right now. So today I want to get into, we're going to focus on inheritance. And I just want to get everything all set up for you. Just make sure I can see who else is on. So we're going to talk about inheritance and how traits are inherited and passed on to the next generation. Last week, we explored cell. We took a journey through the cell, and we talked about the different organelles, the different life processes that takes place with those organelles. We then spent time talking about the chemical reactions that takes place inside the cell itself to help it to maintain homeostasis. Now remember, the cell is the basic unit of structure and function for any living thing. So that means the function of any of the body systems, it's important that you understand how the cell works in order to understand how those systems will work. And so we're going from understanding the basic cell to the fact that cells come together to form tissue, looking at the tissue, and that tissues come together to form the organ in the living thing. And organs will work with other organs to make up the organ systems found in the organism. So now that we have established that, this time we're going to talk about traits and how they're passed on. And I want to start today's session going back to the basic structure of the cell. In order to know how traits are inherited, it's important that you understand how the cell function and work in inheritance. Okay, so let's take a look. I have a presentation here. All right, so this section, remember, we're going to go through the rules of inheritance. So the major topics that we're going to cover today is cell division and genetics, as far as how go through mitosis and meiosis. We'll look at the difference between sexual and asexual reproduction, molecular genetics, bacterial transformation, protein synthesis, mutation, Mendelian genetics, and gene expression. So that's our goal for today's class. So we go back and review the cell. Now in the cell, the nucleus is in charge of maintaining cellular activities. And so here in this diagram, we're going to take a closer look at the cell itself, at the nucleus itself. So I've isolated it from the cell so that we could take a closer look. So I put on my glasses to take a closer look myself. All right, so here we go. So the nucleus, you could see inside of the nucleus is the nucleolus. So here embodied inside of the nucleus is the nucleolus. We're going to see that the nucleolus will be responsible for RNA synthesis. When it's time to produce a protein, we're going to see that transcription will take place here in the nucleus. Then we have the entire thing represent the nucleus. And here, just as how there's the cell membrane, there's a nuclear membrane, which is referred to as the nuclear envelope. And same as saying the nuclear membrane. So things, if you remember what the cell membrane did, it allows materials to move in and out of the cell to help to maintain balance. So we're going to see that the nuclear membrane also allows things to move out of the nucleus. And so in the same manner, it has to be selectively permeable. We don't want all the materials that's inside of the nucleus to be able to leave out of the nucleus. There's certain things that if it leaves the nucleus and end up in the cytoplasm, the pH of that environment, the enzymes that's found in that environment could destroy the genetic information. Also here we see the nuclear pore that allows materials to pass in and out. Found inside of the nucleus is our chromosomes, and the chromosomes is what we inherit from our parents. If you're human, you inherit 23 chromosomes from mom and 23 chromosomes from dad. Different species have a different number of chromosome. The chromatid are uncoiled, like single strand of genetic information, whereas the chromosome is condensed form of that chromatid. And so we're going to see that the chromosome, when uncoiled, is made up of DNA, and the DNA has your characteristics on it, which are your genes. So the information found in the nucleus, determines what proteins your cells will make, and we need proteins for different things throughout our body. We need proteins for enzymes, hormones, different components of our blood is made up of proteins. So we're going to need proteins for different function in our body. So we also see that the information inside the nucleus determines which protein will serve as the enzymes in our body. And as we explored the last time, we saw that enzymes is an intricate part of the metabolic processes that will take place in the cell. Next, it will also determine which chemical reactions will or will not occur in the cell. So we'll also talk about some malfunction when there's mutation that will affect that specific protein from being made. So that's a quick review of the nucleus. Let's move on. Here I have a quick video. When we take a second and listen in.
Each chromosome contains many genes. Each gene on the chromosome contains both exons and introns. The exons contain the coding sequences, and the introns contain the non-coding sequences.
Okay, so I just want to take a look here. So what we're showing you, here's the chromosome that we saw in the nucleus that we just pointed out in the image below. And remember I said that the chromosome is a coiled up DNA. So it's showing you that the DNA strand makes up that chromosome. And what you can also see here is that your gene, there are different genes expressed along different areas of that DNA strand. So different parts code for different characteristics that the organism have. And so to take it a step further, you have these portions in between. And so those portions are referred to as intron, and the intron are the non-coding sections, while the exons, those are the part that's going to code for the characteristics in that organism. This is a great picture that it breaks it down and you can look at them parallel to each other.
Each chromosome contains many genes.
All right, so I just want to skip past this a little.
Each gene on the chromosome.
Okay, here we go. So now moving on, let's take a closer look at the DNA structure. So DNA stands for deoxyribonucleic acid. It's broken up here. You have your deoxyribonucleic acid. That's your DNA. And so the DNA will transcribe, will be there for transcription, to transcribe the genetic information that you need to make proteins. Let me finish that off by saying transcription is going to help to form that RNA that's needed for protein production. You have your hereditary information that's being passed on. It is your genetic code for everything that will be expressed in the organism. And all the development process of the organism is found in the DNA code. So as we take a look at this other image that I have up here on the right, here's the structure of the chromosome again. It's important that you understand the structure of the chromosome and the different parts to it. And so if we take a look, the chromosome, there are two sides to it. And so one side is referred to as a chromatid. So when you have them paired up, you have inherited from mom, inherited from dad. They're referred to as sister chromatids. Holding them together in the center here are the centromeres. And that component is broken down where you have the microtube and the chenochord. And you have your telomeres that are found at the bottom. So these are a description of the different parts of the chromosome. So once again, the DNA is the hereditary material that will be passed from one generation to the next. The genetic information is used primarily to make proteins, and those proteins are used throughout the body for different things. So here, if we were to look at the simplest part of the DNA, DNA is a nucleic acid, and the basic or subunit of nucleic acid is a nucleotide. And so for DNA, the nucleotide, I'm going to use the structure on the right to represent the nucleotide. If you were there at the review session, we spoke about that 5-carbon ring representing part of a carbohydrate group. So this is a sugar, and that's the 5-carbon sugar is deoxyribose. The end in -ose is a good indicator that something is a sugar. Attached up top is your phosphate group, and along attached here are your nitrogenous bases. And so each nucleotide will differ based on the nitrogenous bases. So for example, nitrogenous bases here, you could have adenine. Oops, let's try that again. Adenine. You could have thymine, guanine, or cytosine. So there are 4, I have them listed here. There are 4 different types of nitrogenous bases. And these 4 types is what differentiate each nucleotide, DNA nucleotide, from the other. They also pair up according to what I have shown below. Adenine will always pair up with thymine, guanine with cytosine when we're looking at the DNA structure. So those bonds will perfectly bond together using a weak hydrogen bond. And so when you're replicating, when you're making a copy of the DNA, we're going to have to split that weak bond between the nitrogenous bases and allow for enzymes to create the ligand, or in comparison to the lead-in strand, and cause that new strand of DNA. So if we're going to talk about one cell creating another cell, going through cellular replication, where you need to have from one cell that needs to be created into another cell that's identical, you're going to see that you're going to have to copy that DNA structure. So there's an unzipping process, which means the weak bonds will break along the bases I just showed you, and the new strand will form along. I believe I have a video here. Let's check it out and take a closer look at that process.
DNA replication is a fundamental process by which multiple copies of DNA are formed. The double-stranded DNA is separated by the enzyme helicase.
All right, so I just want to pause here so we could take a look in to see that here's the DNA strand, and even the process of splitting those bonds, you need an enzyme to cause that. And in this case, we have helicase, which is the enzyme that will split the weak hydrogen bonds in DNA. Let's continue.
DNA replication is a fundamental process by which multiple copies of DNA are formed. The double-stranded DNA is separated by the enzyme helicase. Single-stranded binding proteins keep the strands separated. In the leading strand, there is a continuous synthesis of the new strand, while in the lagging strand, there is a discontinuous synthesis in the form of Okazaki fragments. DNA replication is a fundamental process by which strands separated.
I wanted to show you those strands.
In the leading strand, there is a continuous synthesis of the new strand.
Okay, so let's take a deeper look at this section. So the leading strand, this is the strand that's being replicated. And so the strand, the part of the DNA strand that's being replicated, it's being replicated from the 5' to the 3' section. This is controlled by an enzyme referred to as DNA polymerase 3. And so the leading strand is the template that's being used. So if this was nitrogenous base A, this is T. If this is C, this is G. If this is G, this would be C, and so forth. And it carries all the way out. Think about the parent sequence: A with T, T with A, G with C, C with G. And so this process continues. So remember, the DNA has two sides to it. So what happens, what the video is going to show is, once the top portion is being replicated, what's happening to the lagging strand, which is at the bottom, the part that we are not looking for the new information. And let me see if I could get this going.
While in the lagging strand, there is a discontinuous synthesis in the form of Okazaki fragments.
Right, and so that's the bottom section. That will be discontinued.
RNA primases create an RNA primers, and DNA polymerase 3 facilitates the synthesis of the new DNA strand.
All right, so this section might be getting just a little bit ahead because later I want to come back to where it talks about that RNA polymerase. And we're going to talk about when it's time to transcribe into an RNA, that's when that's going to come into play. And you could see here, the new strand is also being formed here. That's going to close up the bottom. So you'll end up having double strands, two identical strands of DNA where we started with one strand. The enzyme allowed the DNA to split, and on the opposite side, you have new strands coming in, ending with two identical DNA strands.
The DNA polymerase 1 replaces the RNA primers with DNA. Finally, the Okazaki fragments are linked together by the enzyme DNA ligase.
All right, that was a great video because you get to see the animation. Remember, these videos are located on Learn America website through the LIU link. So please take some time to review those. So here, let's do a quick practice before we get completely deep into this. Let's just do some basic practice. So my first practice question: hereditary information is stored in genes. So think about that, which are made of, so think about the genes before I add those circles representing the exon, which is the information that codes for the gene. Those are made up of, you have to think about what are they made up of, and we could replicate them, and it also controls the production of. So if you remember on the strand, we had the exons and the introns, and so we saw that the coded area, those exons, they make up that long strand of DNA. And then we just viewed that DNA can be replicated, and they control the production of proteins. All right, and so in box A should have been DNA. It says, what type of molecule in box A serve as a template? And so we said that DNA replication would take place. And which molecules represented in box B? That would be our proteins. So by analyzing the diagram from start, that's going to help you to come up with the answers before you even like really go through what the question is. If you're able to look at the diagram and analyze and try to see if you could fill in the blanks on the diagram, that's going to make you more confident when answering the questions. And this is what we do during the review sessions. So you want to make sure you log in for that. All right, let me just see if we have any more. All right, so here's another one. Remember our rule that A pairs up with T? A pairs up with T, G pairs up with C, and vice versa. T pairs up with A, C with G. So it says that 15% of DNA sample is made up of, 15% is made up of thymine. What percentage would be made up of cytosine? I'll give you a second to think that one through. Okay, let's take a look. So let me just make sure. Give it a second. All right, power cord fell out. Let's continue. We've got power. Yes, we're back in business. So here we go. So that should have given you ample time to go through this. Let's see how you did. So if A pairs up with T, and they're telling you that 15% of the strand is made up of adenine, so that means 15% has got to be made up of thymine on the DNA strand, because wherever T is, A's got to be there. So if I'm telling you that 15% there are 15% of thymines in the DNA strand, we can then assume that there's 15% of adenine there. So the question didn't want to know what's the percentage of adenine? They wanted to know what's the percentage of cytosine. All right, remember C joins with G. So what you would do then is, let's say 15 and 15, so we could say that's 30%, 30% of the DNA is made up of adenine and thymine, which then leaves us that 70% would be made up of C and G together. But they don't want to know, the question is not asking what is C and G, the total. They just want to know a C. So what we could do then is divide that in 2, all right? And that gives us 35%. So then we know that 35% is made up of cytosine, 35% is made up of guanine. And so if you add that all up, you should get your 100%. And so that's just a question that's reviewing the bonds that are formed in the DNA, that A pairs up with T, T with A, G with C, C with G. Good, I hope you got that one right. All right, let's move on now. Outside of the nucleus is still very relevant to this topic, but I'm gonna move over now to the ribosome. So we're gonna move out of the nucleus. Remember the ribosomes, you could find them attached to the endoplasmic reticulum, which makes up the rough endoplasmic reticulum. And we also find them free floating through the cytoplasm. So let's take a closer look to the ribosome. So here, the ribosome, there's actually 2 parts to it. Most of the time we just see it as this dot in the cell, but there's actually 2 components to it. There's a large subunit and there's a small subunit. So here's the full image of the ribosome, but there are 2 sections to it. The larger subunit is illustrated here. And so there are different sites in it. There's a transfer RNA site, P and A site that's found in it. In eukaryotes, there's 3 rRNAs, which characterize maybe 40 proteins. There's an active site found in that top portion. And whereas in the smaller portion, there's a difference in the structure itself. And so here we'll see in prokaryotes, this is what you would get. Whereas in eukaryotes, there's the difference. Okay, so yeah, I don't want you to just look at the ribosome just being this dot, but it's a very complex structure which has a top and a bottom that carry out different function when making proteins. So that was a quick overview. Let's continue. So the organelle in which amino acids produce to form proteins is the ribosome. And so here's just a quick overview of how that would take place. So I just want you to get, this is the big picture. So there's the DNA, the nucleus. And inside the nucleus, we established that the DNA is found in the nucleus. The DNA is going to get transcribed into a message because the DNA cannot leave the nucleus. If it leaves the nucleus, there are enzymes in the cytoplasm that would destroy the DNA. So the DNA needs to stay in the nucleus. So it transcribes the message, whatever the protein is or the amino acid that needs to be made. The DNA, that portion of the DNA gets transcribed into a messenger RNA. The messenger RNA has the ability to leave the nucleus, travel, so it gets transcribed, and it travels to the ribosome where it will get translated into amino acids. Those amino acids will combine to form proteins. So I like this picture because it's just this image because it gives you an overview of the big picture of how protein is being synthesized in the cell and the role of the nucleus in protein synthesis. And so as we're going to talk more about the transcription process or making of proteins, it's important that we also understand the RNA. And so RNA nucleotide helps to transfer information for protein synthesis. So I specifically spoke about the messenger RNA in the previous image. Let's go back to it. That's where the DNA gets transcribed so that it could leave to take the message to the ribosome. But there are 2 other RNAs. We have the transfer RNA, which we'll find in the ribosome. And there's the ribosomal RNA, which I just briefly showed you that we would find it in the subunits of the ribosome. Let's continue. So now we've seen this image before when we look at the DNA nucleotide. But the image is similar for the RNA nucleotide, as in here is a 5-carbon sugar. There they are. We discussed this in our review session. So that tells you it's in the carbohydrate family, it's a sugar, but this time it's not deoxyribose, it's just ribose. The phosphate group still there. And the nitrogenous group is still here. But there's one difference. Before, let's take a look over here at the different combination. So we see adenine is the same as the same base that we saw in DNA. We saw guanine and cytosine. But notice we do not have any thymine on the RNA component, on the RNA nucleotide. That T is replaced with uracil. And so uracil will now bond with adenine, since there's no thymine, and G and C still has the same relationship. So that's the only difference here, is that, not the only difference in the bases, is that thymine is not there, but they function in the same way as in there's a base pair that takes place. And instead of having deoxyribose, we have ribose as the sugar. So if we were to compare the two, DNA, here's the DNA strand here on this side, and we have the RNA strand on this side. And so DNA, we have deoxyribose compared to ribose. We have thymine instead of uracil. A pairs up with T, whereas A pairs up with U. DNA, it's a double strand. So if we take a look here, it's a double helix structure. Helix, it's going around, but there's two parts to it, and we're using the pink and the blue to represent the two strands that make it up. Whereas in the messenger RNA, it's a single strand. And so that single strand, we could see the uracils found on it. And this single strand will then travel over to the ribosome where it will be translated. All right, I hope this gives you a good visual of the two. So transcription. Transcription is the copying of the genetic information from a strand of DNA into a molecule of messenger RNA. So let's take a closer look. Actually, before we do that, translation, the point at which it translates is using the messenger RNA molecule to build a protein. And this occurs inside of the ribosome. And here's the messenger RNA. Let's take a look at this image. Single strand. And we're going to see that every three represent a codon. So if I was to interpret this messenger RNA just by writing the letters down, this image is an interpretation of that ribbon. So we could say each base that's along here represent one of these letters. Just let you soak that in for a second. So the first three bases here, so maybe 1, 2, can't see the third one, 3, could be a G, C, U. The next three, 1, 2, 3, could be A, C, G, and so forth. So the different types of amino acids, these are them, and this is it. These are the types of amino acid that's found in everyone. So if we take a look, how many? We have 1, 2, 3, 4, 5. Sorry, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. Did I do this one already? No, 19, 20. I have some duplicates. All right, so we'll see that different codes, more than one code, can code for a specific amino acid. So here, the code CGU, CGC, CGA, CGG can code for the same amino acids. And so these are the codes that's found in everyone's body. This is it. And so the way that each code is sequenced, that determines the type of protein, and that's what makes us different than each other. If you think about it, if they're the amount of letters that's in the alphabet. Think about how many words can you make from those letters. And then think about how many sentences you can make from those words. Oops, let's go back. So it's the same thing, even though there's a limited amount of amino acids. When the DNA sequence is transcribed and translated, there are set amount of amino acid, but the way they form with each other, you can create numerous amount of combination, which lead to why we're all so different from each other. All right, so the translation process, here's another image. So here's the ribosome. Remember, there's the bottom, small unit. Here's the large unit. Here's that messenger RNA. Number 1, represent the messenger RNA. And so that messenger RNA is now over to the ribosome to be translated. And then what we're going to see is these little, say they're almost like upside down T's. They're reminding us these are the transfer RNAs. And they're going to transfer over the amino acids that we're coding for. And on the transfer RNA, for example, this one at CCC, that represents the anticodon in comparison opposite of our code. So if the messenger RNA code that it brought to the amino acid, to the ribosome is GGG, it's going to be translated into, for example, this amino acid glycine. So the way that the transfer RNA will know what amino acid to bring, it's going to have to pair up with the anticodon, which is CCC. So CCC is the anticodon to the tRNA that's associated with glycine. So there's a matching combination to take place. So let's try that one more time. Messenger RNA code, every 3 codon codes for an amino acid. Once it gets to the ribosome, the transfer RNA will match based on the anticodon, which in this case is CCC. And so that transfer RNA will know that it's correctly matched, and it will then be linked to that amino acid. The process continues on and on, so you will see amino acids bonding together, like we see here in section 3. We see numerous amino acids bonding together, and that's what's forming a protein, because amino acids make proteins. Let's take a more in-depth look at this by watching this video.
The stages of translation are divided into initiation, elongation, and termination. Eukaryotic mRNA has a unique 3' end with a poly A tail and a methylated cap at the 5' end. In the initiation step, the mRNA binds with the small ribosomal subunit. The second step is elongation, which requires the presence of tRNAs. tRNA molecule contains an anticodon, which is complementary to the codon found on the mRNA. The mRNA start codon is AUG, which corresponds to the amino acid methionine, which is attached to the appropriate tRNA. The large subunit of the ribosome now binds with the smaller subunit to create the peptidyl or P site and the amino acyl or A site. The first tRNA occupies the P site, and the second tRNA occupies the A site. Methionine is joined to the amino acid that is attached to the tRNA in the A site. Once this happens, the free tRNA occupying the P site can now exit. As the ribosome moves along the mRNA, each amino acid attaches to the next amino acid in the A site. The elongation step continues until the tRNA encounters the stop codon, and then a releasing factor binds to the A site. Translation is now terminated. During termination, the ribosome dissociates and the newly formed protein is released.
Wow, that was a great video. And you got a chance to see how the P site and the A site came into play. And one of the things I'll go back just to emphasize is, in addition to the name of the amino acids, you do have codes for stop. And so you'll find that code along the messenger RNA. And so that's how the ribosome will know when to stop making that protein. I think that video is right on point. Remember, you could always go back and review that on your own. So now we want to go and discuss what happens now if there's a change, or what happens if something changes the code to something else. How does that affect the type of proteins that will be made? And so we refer to any kind of change in the genetic code as a mutation. And so mutations tend to have a negative connotation to it, but mutation can also have a positive effect. So there are negatives and positive effect of mutations. All right, so let's take a look at some different types of mutation. So we see here transition referred to the replacement of purine with another purine or replacement of a pyridine with another pyridine. What's that all about? Let's take a look. So here's the original strand of DNA. And just by looking at the original strand, the original strand of messenger RNA. So here was the message that was transcribed from the DNA. Here is the strand. And now you could see that every 3 codes for a particular amino acid. So everything has been done, it's been transcribed. And so what it's saying is, if this C cytosine, a mutation occurred to change the C into an A, we know that CAA originally codes for this particular amino acid. But if it's changed to AAA, it will change the type of amino acid that's made, which will then change the protein that's produced. Most of the time this happens, but it doesn't happen all the time. Sometimes a change will occur and you're still able to make the same protein. But in this case, we're seeing that there was a base substitute. C was substituted by A, which then changed the protein that was made. So this is base pair substitution. So that's one type of mutation. How about this one? Let's take a look at the same example. Here's CAA. Oops, lost my screen. Let's go back. CAA changes now. There's a single base change. So this is transversion, referred to the replacement of a purine with a pyridine. We're talking about the different types. So for example, we have C. Remember, C pairs with G, A with T. Let's keep that in mind. All right, so we're here seeing that the C is changed to a G, which is a pyridine in comparison to the previous one. A and C, their structure refer to them as purines. So it's a base change of the same, let's say, grouping. Whereas C and G are pyridine, C is a purine and guanine is a pyridine. So here it's change. Transversion is where you're replacing it with an nitrogenous base from a different group, which is the pyridine. And so frame shifting, here's another example. So frame shifting, let's take a look at our original strand. Here's the DNA strand. We're going to focus on the C, which is in blue. So if I can zoom in. Oh, yep, here, let's zoom in. Here's the C. We could see that it was translated. Here are the amino acids that's translated. Now, if we were to insert a single nucleotide, so from here, instead of in this space here, in between the two, between the black T and the C, blue C, if we were to insert the letter A, that would then shift all the messages thereafter. Because remember, you have to read every 3 codon. So before, the message was CAT, but now that we've inserted a single base, now the message changed from CAT to ACA, which in turn produced a completely different amino acid. And that then throws everything off. Everything else on the sequence changes. So now, whereas this before was also CAT, The next three is now TCA, TCA, and so forth. All right, so an incorrect amino acid, which may produce a malfunction in the protein. So we could have it in a situation where we saw before with the purine where it may not cause a change, but we could see where it completely might changes the protein to where it cannot function. Let's look at another example. Here, in this case, we're going to delete C. Remember our the original strand, C-A-T. So let's take a look. What if we were to take the C out? So by taking the C out, it shifts from CAT to now ATC, ATC. And in this case, it just so happened that it makes the same amino acid. Here's another example. Here, if you drop down, the original code for the amino acid sequence is here. You'll have that code, the CTC, codes for stop. All right, so remember what the original was. Sorry, let's clear that up. And then we deleted one. So instead of it being the same amino acids, this is the after effect where it caused it to stop. So you're no longer able to produce that protein. All right, so this was one example. Kind of spoke a little too soon on that protein production. So just make sure we have this right. Here, it's showing you that C was deleted. This was the original amino acids that should have been made, but now that C is deleted, it turns CAT into ATC, which is a stop code. So that protein will not be made. And so the act of shifting the information over is referred to as frame shift mutation. All right, just in time for a practice question. All right, so this one kind of touched on our last session, but it's applicable to what we're doing today. Take a look at this. Remember, try to fill in the diagram before even looking at the question. So we see block A, B, and C. So it says identify the type of building blocks that's represented by A, B, and C. So if we take a look, the building blocks, they're going to come together to form proteins. So we should know already what forms protein, and that would be amino acid. So the answer for number one is amino acids. And so what effect would happen if we change the sequence? What effect would it most likely have on the protein? It would change the kind of protein that's being made, or it would probably stop the production of that particular protein. So those are options. So it's most likely it would change that protein. It will make something else. Here's another question. Let's take a look at this one. What's the relationship between these three structures? Let's zoom in. Oops. Zoom back in here. What's the relationship between these three? So you should be able to tell the DNA is found, oops, I gotta zoom back out. So the DNA is found inside the nucleus. The DNA sends the message to the ribosome, and that message would be to make proteins. Proteins are made in that ribosome. All right, that's the connection. Good. All right, so we're getting ready to move on to our next section. So we've finished the basic DNA the importance of the nucleus. We've looked at the cell, and we're going to build up on this for today. We're going to move into reproduction and inheritance. So we want to finish this half, the cell component, maybe in another 5 minutes, and then we'll take the second half of today's session looking at how the whole genes and how genes are inherited. So here we go. The centrioles now are found in animal cells alone, and they are responsible for the spindles, producing spindle fibers that are an important part of cell replication. So let's take a look. One type of cell replication is mitosis. Mitosis is when you make an identical copy of the cell. So mitotic division, I have an example in the corner here on your right side, that if I have an organism, I have a cell that has 46 chromosomes. In mitosis, you're going to see that the cell will split. Remember, the DNA is going to split apart, controlled by enzyme. You'll end up with 2 identical copies of the DNA. That's what's happening here. And so you'll end up with 2 cells that are identical. And so mitosis is when a cell genetic material divides and replicates, creating 2 complete cells of identical genetic material. A cell division that results in 2 identical daughter cells. And so there are different phases that you go through when you're doing mitosis. And the first phase is referred to as interphase. And this is the growing process. This chart down below give you a perfect example of it. So let's take a look at it. This entire section in purple refers to the interphase. And so in interphase, there's several processes that takes place. It's really referred to as the resting phase because this is when the cell is growing. There's growth and DNA. The DNA is being replicated during this time. The cell starts preparing itself for replication. And so even though it seems like there's a lot going on here, the cell is not actually dividing. So that's why it's referred to as the resting phase. And then after that, we have mitosis, where it goes into prophase, metaphase, anaphase, telophase, and lastly is cytokinesis. So let's take a minute and take a video run through of each section. Okay, maybe I'm not ready for that video. Oh, nope, I just want to talk about it, which works. So prophase, we're seeing in prophase here. Prophase, you're going to see the nuclear membrane is going to be broken down. So you're going to see the chromosome themselves, or the chromatid, they're going to start to thicken at this initial phase. Now, let's get the video. Oh, what's going on? Oops. All right, no video. Oh, no, I got it.
Mitosis is the process in which a eukaryotic cell divides into 2 cells. The 2 daughter cells contain exactly the same number of chromosomes as the parent cell. During new cell formation, the period of cell growth is called interphase. During this phase, the DNA is replicated. Prophase is the initial stage of mitosis.
And so you want to see, as replication is taking place, you could see these guys here, the centrioles, starting to move away from each other. We're used to seeing them right next to each other in the cell, but now they're moving into action. This is what they're there for. So they start to separate, and they're making their way. One's making its way to the opposite end. But also take a look as to what's going on inside of the nucleus. So we see that the chromatids are condensing into the chromosomes. Pretty cool.
During prophase, the nucleolus disappears and the chromatin condenses to form chromosomes. The centrosomes start to move towards the poles, organizing the spindle microtubules between them. By the end of prophase, the nuclear membrane breaks down and disappears. During metaphase, the spindle microtubules attach to the centromeres of the chromosomes, and the chromosomes line up along the metaphase plate.
All right, so as you could see now, we're moving on to the next phase, metaphase. They're in the middle. The metaphase plate is aligned in the middle. So metaphase, it's in the middle of the cell, where the chromosomes will then get attached. The spindle fibers will attach themselves to the chromosome in preparation for separation.
Anaphase starts with the contraction of spindle microtubules, which split the chromosomes into sister chromatids.
All right, let's check that one out too. So you could see how amazing is that the spindle fibers, now that they are attached, they're going to yank each sister chromatid to the opposite side. Remember at the beginning of today's session, I showed you that the chromosome had two sides to it. They were referred to as sister chromatids. And so now we need to separate, and we're going to send one set to one side of the cell and one set to the other to be duplicated, to represent duplication.
The chromosomes start to move towards the poles. During telophase, the chromosomes reach the poles and the nuclear membrane is reformed. Cytokinesis begins with the formation of a...
All right, so before we go on to cytokinesis, you're going to see, once again in telophase, you're going to see the nuclear membrane will come back and reform. And you should have identical number of chromosomes on each side. Cytokinesis now is where you're going to split the cytoplasm, pretty much, and so that you could have even amounts on each side. And remember, it's when it goes back into interphase again, that's where you're going to see an increase in the genetic information is going to get duplicated again. The cytoplasmic material, the whole preparation for division will occur all over again. And the process continues over and over.
Cell plate. Following cytokinesis, two daughter cells are formed that are exact copies of the parent cell.
Okay, so that's how you go from having one cell to two cells that are identical. All right, so now let's talk about fertilization. The fusion of the two nucleus, the sperm and the egg. So the sperm has its own genetic information. Egg has, I would say, half the genetic information of the dad. The egg has half the genetic information from the mom. We're going to see where they're going to combine. So we're going to use N to represent the haploid number of genetic information. And it's going to help to form diploid number of chromosome, meaning a full set of chromosome information. That fertilized cell is referred to as a zygote, and it's a cell that contains all the genetic information needed by an offspring. And so what you could see is once the sperm and the egg meet, here A is your zygote. And then you're going to see the cell goes from one into two cells. How will it do that? Mitotic division. So that's where it's going to go through interphase, prophase, metaphase, anaphase, telophase. You'll end up with two identical cells. Because how do you go from one cell to being made up of so many cells we can't count it? So it's going to go through this rapid cell division. Then from B, it goes on into two cells into four cells. How does it do that? Each one of those cells will then go through interphase, prophase, metaphase, anaphase, telophase. And then you end up with 8. Then those 8 divide again. And then they'll divide some more, and more, and the process continues. So there are different phases that this fertilized egg will go through when we're creating the embryo. And so this initial mitotic phase that takes place, in development of the embryo is referred to as cleavage. And let's take a look at cleavage.
After fertilization, zygote divides into tiny cellular units. This process is referred as cleavage. Zygote undergoes mitotic cell division to multiply. Here, the cytoplasmic growth is arrested. So, the cell size will not increase, but proliferate in their count.
All right, that was an important part. So as we're saying that the cell is dividing and making a copy of itself, don't think that the cells are getting bigger. Each time they're getting smaller and smaller. Right, so as it divides, that cytoplasm divides, it divides and the cell is going to get smaller. And then it divides a second time. Think about that same amount of liquid that's in it. It's going to divide down. So the cells, there's more of the cells, but the size of the cells are getting smaller to the point where we were one cell and now we're made up of trillions of cells that we can't count. So we can't see our cells with our natural eye. It's because there's so many that they've become so small.
Cleavage process produces blastomere. Blastomere form blastula in the end and leads to gastrulation.
Well, I wonder if I could get back that section. Let me see if I could.
After fertilization.
Yeah, I want to take a quick look at
cleavage
this.
Process.
This again. So here we have the ball of cells that's created. So this is when you're creating an embryo. You went from the one cell, it went through cleavage, now we're at the blastular phase. And a good way I like to think of the blastular, but pretend you're going to blast a hole in this ball.
Produces blastomere. Blastomere form blastula.
See, take a look. So it's almost like you blast a cell, blast or opening in this ball. And so when it's open, what that does is increases the surface area for more cells to develop. And so we're going to have some cells on the inside, some on the outside, and some in the middle. And that's going to give way to the different types of cells
in the end and
in your
leads
body.
to gastrulation.
All right, so
after fertilization,
let's do that
zygote
again.
divides into tiny cellular units
if I could get it.
in the end and leads to gastrulation.
Not fast enough. Yes, I got it this time. All right, so that ball that we blast out, let's take a look. There's an outer portion to it. So you have these cells that's on the outer portion of the ball. Then you have the cells that are here, and then you have these cells that are here. So you have 3 layers of cells now that started from that one cell. Now there's so many cells that they're going to divide into 3 sections. And these 3 sections are going to give way to the different parts of the embryo. We're going to see the ones on the outside, they're going to give way to like your skin. The ones that's on the inner, they're going to give way to like muscles and organs. And you'll see the other ones will give way to like connective tissue. So it's important that you see this, that the different layers are going to contribute to the different parts of the body. Good. All right, so that process is referred to as differentiation, where you went from having that one ball, the zygote, and it changes or divide into different sections, where you're going to have some sections making your skin, some will make your nerves, some will make your muscle cells. That's called differentiation. So differentiation is a process of developing cells with special function and structure. These cells maintain identical genetic information. So your skin cell, your nerve cell, muscle cells, they all have the same DNA in them. It's just that different portion, different parts of like the exons are being expressed into whatever the function is. So the location of these cells, is it in the ectoderm, the mesoderm, or the endoderm, what layer it's in is going to determine what type of cell it changes into. So I have a few pictures in the picture below. You could see if I could enlarge this a little bit. Those cells that are found in the ectoderm, that's the outer portion. That's going to give rise to your skin cell, nerve cell, the pigments in your cell. It's going to give rise, also you see your gastrula, the sperm cell, egg cells are going to be contributed to that area. Where we have the mesophyll, which is that middle layer of that circle I just showed you. And that's going to give rise to now your muscle cells, like your skeletal smooth, your blood cells, your cardiac muscle cells are going to be developed in this area. And the outer layer, the most inner layer, which is referred to as the endodermic layer, that's going to give rise to your lung cell, thyroid cells, for example, pancreatic cells. That's a few examples of the different parts of the body that's made based on that differentiation that takes place in the circle. So they all have the same genetic information in it. It's just that different parts of the DNA is being transcribed, or sorry, being expressed to give you the different types of cell. So the environment can also influence gene expression. So nutrition, temperature, sunlight, these are all some factors that can affect gene expression. And I'm not just talking in humans, I'm just speaking of living things in general, that all of these can affect the way genes will be expressed, what they'll become. And there are just a few examples like the chlorophyll, light, the sunlight will affect the chlorophyll, the color of the leaves. The environment, identical twins having the same genetic information, two different environments can end up looking, appearing differently. And we see that stem cell, those uncoded cells, we could manipulate them to become what we want them to do or mature into. Here's a practice question. Let's take a look at it. So we could see here there's two different environments. One's 20 degrees Celsius, one's 31 degrees Celsius. Humidity is 20% versus 95%. And the genes are represented here as genotypes AA, big A, small a, small a, small a. And it's showing the phenotype as in red, red, white, whereas in 95 degrees, those same genotypes are expressed as white, white, white. So what conclusion would you draw from this? Right, so the conclusion should be that the environment will influence the gene expression. We could see in this organism that the temperature influenced what color would appear. And so remember, the cells are going to give rise to the tissues that we just saw, the skin, your nervous tissue, your, what else, muscle tissue. That's all going to give rise to the organs, and your organ to your organ system. I love checking out these videos. Hopefully I have one more for you here. Let's see here.
An animal body consists of 4 types of tissues: epithelial, connective, muscular, and nervous tissue. Both internal and external structures of the body are surrounded by epithelial tissue. Protection and absorption are some of the functions of these tissues. Connective tissues bind organs together, form blood, and store fats. Muscle contraction and relaxation occur with the help of muscle tissues. The skeletal and cardiac muscle tissues have different characteristics, but perform similar functions in bones and in the heart. While the nervous tissues facilitate conduction and the integration of stimuli, they also protect and support nerve cells.
So that's to show you the different cells that were formed because of differentiation. Those will then put together to form tissues. Tissues make the organs, which then function in a system in the body. It's pretty straightforward. And we'll see that all the systems will work together to maintain homeostasis in the organism. And in our last 2 sessions, lecture sessions, we're going to break down each one of these systems So hopefully you guys could join me then. But everything links back to the circulatory system, which helps to pretty much deliver what's needed and also to pick up materials that's not needed to help it get out of the body. Like we could see nutrients will be absorbed into the bloodstream. Where in the excretory system, we see that blood gets filtered in the kidneys, which is a part of the excretory system. Gas exchange takes place between the blood and the alveoli of the lungs. In the endocrine system, we see that the hormones transfer from the glands by way of the blood to get to their target tissue. Muscular system, gases and nutrients from the bloodstream is going to aid in the production of energy molecules such as ATP. And we see our white blood cells create an immune response whenever we're been exposed to pathogens. And so once again, we'll get into more details of this, but here you could see two systems and you should be able to tell how they're related or how they could connect to each other. We'll come back to this today, if time permits, as a quick review. Also, I didn't want to go ahead without giving you a review that we've been talking about animals, but plants also have some of the same characteristics, as in transport takes place in their body or in the structure through the xylem and the phloem, which we mentioned in last session. The xylem transport water throughout the plant. Phloem transport nutrients throughout the plant. The leaf has openings which allow gas exchange to take place, and the guard cell is going to regulate the opening and closing of those stomates. All right, so let's now talk specifically as to how these traits are passed on. So traits are the characteristics, and remember the genes are going to determine. Alrighty. Oh no, it's just my picture. All right, so here we go. I have an example of a chromosome here. And so on the chromosome, we have different, just a reminder to show you that there are different genes or different traits that are found on the chromosome. So we have Mendelian's law of inheritance. So there's the law of segregation. All right, we're going to talk about how traits are inherited and passed on in details. Let me just double check to see if we have any questions. Let me just look and see if there's anything in the chat. All right, and I'm still seem to be going. All right, so let's get back to our presentation. Okay. All right, so we're moving into the last section. So I just wanted to check to see if there were any questions before I just keep going. Oh, come on. All right, so in our last sections, we're talking about laws of inheritance. So one of Mendel's, if you don't remember, Mendel lived in a monastery. He was a monk, and he studied inheritance using pea plants. He used pea plants because they were easy to use. You're able to see the next generation quickly compared to another organism. So it was very efficient for him to use. So he came up with the law of segregation, which says that genes, you're going to hear me start using the word alleles, separate from each other during cell division, such as meiosis. This is when cell division occurs in the sperm and the egg cell. Then we're going to see that they're going to recombine during fertilization, during the combination of the sperm and the egg. This is going to give the characteristics to an embryo based on how these traits are going to combine. So let's keep going. Another one of his laws is the law of independent assortment, meaning that these traits are going to connect randomly. Different traits are going to separate, but when you form a gamete, it's going to be random, meaning that you don't know what trait's going to end up in what sperm cell or which egg cell. Which just remind me, we need to go back and refer meiosis. Let me just double check, see if I have it coming. Yeah, we should go back and refer meiosis. All right, so Mendel came up with an hypothesis. His hypothesis was a pair of genes that carries the same characteristics and they are found at the same location on the chromosome. So here's the chromosome. Let's see if I can get this right. So if hair color is around this region, here's mom's information, dad's information. It's going to be found at the same region. An organism have two genes, one from each parent. So let's say this is from mom, this is from dad. All right, a sperm or egg cell carries one allele for each trait because the allele pairs segregate during meiosis. So you're gonna have one allele for hair color, all right? You'll have eye color, etc. You get the point. When the two genes are paired let's start that again. When the two genes of a pair of different alleles and one is fully expressed, show up, while the other is not noticeable, the effect on the organism appearance, when it affects the appearance of the organism, that's referred to as the dominant trait. Whereas the recessive trait is the one that's not seen, the one that's unnoticeable. But the dominant trait will be expressed and be seen. So if we were just to organize our thoughts, remember the nucleus is the largest. Oh, come on. And the nucleus, inside the nucleus, we'll find the chromosomes. Chromosomes, find the genes. If we wanted to go less than that, that's made of DNA. Go less than that will be a DNA nucleotide. All right, if we wanted to go less than that, that would be our bases, like our sugar, this deoxyribose, our phosphate group, and our bases. That's what we would find here, even smaller. So any slight difference or variation can be caused in sexual reproduction. So remember before I said that mutation could have a negative or a positive trait, but sexual reproduction is a type of mutation. It's a change to the sequence. When meiosis is taking place, there's crossing over of the genetic information. There's some genes that are linked together, so they create new sequences. And that's what gives the child a different characteristics than the parents. So even though you have mom's information, dad's information, you're not identical to them because that combination, that mutation will change and create the new sequence, which is the offspring. And so let's take a look at crossing over. So this is an example of something that takes place in meiosis. This is when let's say mitosis takes place inside the sex cell, like inside the testes or the ovaries. It will occur twice, which then it's called meiosis. So you're going to go through interphase, prophase, metaphase, anaphase, telophase. Then it doesn't stop there in the testes and ovaries. It will go for a second set of division. So it'll go prophase, metaphase, anaphase, telophase. So you'll end up with 4 cells instead of the 2 we saw before. And so one of the things that happen in meiosis, in the cell division inside the testes and the ovaries, is crossing over. There's an exchange in the chromosome information. So you could see here we have those homologous chromosomes. But during the division process, you're going to see pieces of the DNA will change location. So there's a translocation that takes place. So then now the original message, this is now different than this. And this, right, is different than this because it has that new set of information on it. And that lends itself to the new characteristics. Gene linkage, we could also see that there's certain traits that will be linked together. Like if someone has red hair, most likely they have freckles. All right, if another one, if you have dark hair, most likely you'll have brown eyes, things like that. So they're most likely to be linked together. And so to determine the sex of the organism, let's say we're going to use a human organism as our example because we have 46 chromosomes. Remember, 46 chromosomes. And so if we were to pair them up, we would end up with 23 pairs of chromosomes. And so through karyotyping, we're able to lay out the sets of chromosomes. And what we'll see here is the 23rd pair. They're referred to as our sex chromosome because that determines if you're going to be a male or a female. And so those sex chromosomes is written here. If you have two X chromosomes on the 23rd pair, you are a female. And if you have an X and a Y on the 23rd set, that makes you a boy. And so there's a 50% chance of being a boy, 50% chance of being a girl. So the 23rd pairs are referred to as sex chromosome. All the other ones, 1 through 22, are referred to as autosomes. And so there are different types of mutation that takes place on autosomes in comparison to sex link mutations, which occurs on the 23rd sex chromosome. And here are some genetic disorders. So we have sickle cell anemia, which is caused by incomplete dominance of allele. There's a single nucleotide mutation. We saw that before when we reviewed mutation, that there was a single change. So this way, we're talking about inherited traits. So you could inherit sickle cell anemia. There's a change in the nucleotide. What happens is, take a look at this image at the bottom. T, thymine, is changed, mutated to an adenine. And so what happens is that base changes the regular shape cell to be a sickled shape. So instead of it being a nice circular shape blood cell, it now has a sickle shape to it. And that affects that person's ability for gas exchange. Sometimes they'll have suffer from extreme pain because of that sickled shape of the blood cell. And so now DNA is a hereditary material that transfer all genetic information from one generation to the next. For example, there is a presence of a disease in one of the parents in the first generation, it will be transferred on to the next. So in this little image, in this pedigree chart I have here on the right, you could see where we've shown that dad is a diseased parent carrying this particular disease. Mom doesn't. And as they have kids, you could see how the trait is passed on, and it will be passed on to each generation. Another disorder I want us to review is Down syndrome. So now Down syndrome is a non-disjunction of chromosome number 21. So remember I said there are 46 pair. So there's 2 at every autosome number, except when someone has Down syndrome, they have a third, an additional chromosome on that number. And so that's what gives you Down syndrome. And so this occurs more the older a mother is at conception, the older she is at giving birth, the greater her chance of having a child with Down syndrome. And has to do with those spindle fibers separating. When they separate, the older you are, more difficulties in them separating from each other. So we can see that the percentage of having a child with Down syndrome is greater the older you get. But in our society, we have so much technology and so much medicine right now to see this ahead of time, or just diagnoses, or there are all these gene therapy that's taking place. to make us a little bit more aware and just more knowledgeable about this process. Cancer is when there's an uncontrolled division of these cells. So mitosis is occurring out of control. So that's when you'll end up with a clump of cells. So you have like a tumor, you'll have that lump, that cluster of cell will begin. So there are other diseases that I could show you here, like Tay-Sachs affects the autosome chromosome, and that affects the lysosomes. We have cystic fibrosis, which that is affects the osmotic, as in osmosis, effect in the lungs. So there's an imbalance which cause there to be a buildup of mucus in the lungs. And so that affects the breathing process. And then there are some disorders that's linked primarily to the sex chromosome, such as hemophilia, color blindness, and baldness. Here's some of those techniques I'm talking about. Amniocentesis is where they withdraw the fluid from the parent during pregnancy, and you could examine the cells there. Karyotyping, we can use that in Down syndrome because we're looking at the pair. It's a large photo of the chromosome setting. And by looking at that chromosome setting, you can, see 1 second here. Here we go. All right, so here you could get a big picture of the chromosome, and that gives you, you could see the grouping of chromosome to see whether there's an abnormal number. You can use a pedigree chart to trace the patterns from one generation to the next. So using a pedigree chart, you have something similar to this. You have the circles represent female, the square represents the male. And so on the first line, this will show you that these two are married or these two conceived. And the line below represents the next generation. So these are the kids. All right. And so these kids, the traits are then passed on. So mom is referred to as a carrier, and dad exhibit the trait. So you have a key here. And so if mom had the trait, it would be completely filled in if she had it. All right. But because most of these traits are carried on the X chromosome, you'll see a lot of times the moms are carriers because you'll find a trait on one X chromosome and not the other. So if we were to do a Punnett square for this, let's see here. Let me bring down a whiteboard. And so if we have that mom is a carrier, let's put a dash here, show that she's a carrier. Through segregation, law of segregation, we're going to have those chromosomes split from each other, and they're randomly distributed in each egg cell. And I believe we said dad was normal. So dad, X, and Y. So when you do the Punnett square, let's go down and across, down, and across. So we have down, and across, down, across, down, down. Actually, let me change the color of my pen here so you could see what traits came from dad. Here's dad, and mom is in the red. Oops, such a clicker here. Here we go. All right, so If you take a look at this, you could see the red represents traits coming from mom. Let me change that over here also to a purple. All right, so we could see how the traits are inherited. And so mom is the carrier of this disease. She's carrying this trait on her X chromosome. So this could be an example of color blindness, hemophilia, which is a blood disorder also where that person has problems with clotting of the blood. And so what can we tell from this about their children? And so what we could see is it's possible here that they could have one female. I'm going to write them over here. It's possible that they will have a female that is a carrier of the disease. All right, because you have it on one X, it's not on both. So if it was on both, that would make the female, she would have the disease. So that's why you see the circle that's half shaded on the pedigree chart. And then we have, it's possible that this couple could have a kid that is normal, a normal female. So that would be a regular circle. And so there's a 25% chance that they'll have a daughter that's normal for, let's say this is for hemophilic. And it's also possible they could have a 25% chance of having a female that is a carrier of the disease. Then let's look at their sons. It's possible 25% chance that they will have a son that have this disease. Because he's only got one X, and if the disease is found on it, he's got the disease. But there's also a 25% chance that they could have a son that's normal. So these are the possibilities for these two couples. If they were to mate, a carrier mom was to mate with a normal dad, these are the chances, the percentage of them having a kid, a female being a carrier or not, or a male having the disorder or not. So yeah, we just lost connection for a couple minutes there. So let's see. Let's just finish up. We have a couple minutes left. And yeah, let's go back to what we were talking about. I was talking about biotechnology. And let's see if we can finish up without losing our connection again. Okay. All right, so where we were at in our last section here, we were discussing biotechnology. So I'm not sure, let me just go back a little bit. In this section, we were talking about what is biotechnology, and we talk about manipulating genes and traits to pass them on. Then we said that there were different ways that it could be done, and there were some benefits to biotechnology. And then went over talking about the techniques such as artificial selection, where you're actually mating two organisms that have the desired traits that you want. And so the next thing, just before we lost connection, I was getting into genetic engineering. And so genetic engineering is when you're introducing the organism, the gene from one organism into the next. So I went right to the video before we got disconnected.
The plasmid DNA and the gene of interest.
All right, so what I wanted to say was the DNA from the bacteria, there's a ring of DNA referred to as the plasmid. And so over here, it's represented to your right, it's this is explaining or showing that desired trait, whether it's a growth hormone or maybe insulin, that's the genetic information we want to make copies of. That's the genetic information we want to insert into the bacteria. So the process continues.
Are isolated from the host. An appropriate restriction site on the plasmid DNA is identified in order to insert the desired.
All right, so we're going to have a specific code. We're going to isolate specific area. We're going to use a restrictive enzyme that will know where to bind on that plasmid DNA from the bacteria. It's going to open it up.
DNA and the gene of interest are isolated from the host. An appropriate restriction site on the plasmid DNA is identified in order to insert the desired gene. The plasmid is cut at the restriction site and sticky ends are created using restriction endonucleases. The target gene is inserted into the plasmid DNA with the help of the enzyme ligase.
All right, so a lot of
This
people,
hybrid DNA is no.
I need to clear that up. There's not actually glue being used in this process. So when the enzyme cuts the DNA, there are these sticky ends that are exposed, and they're going to complement the new DNA strand that's going to enter in. Earlier in the session, we spoke about A pairing up with T, G with C. So the sticky ends that's exposed, you're going to see the bases exposed. So whether it's A, T, G, C, the complementary strand of the section of DNA you want to insert is going to be the opposite, T, A, C, G. And it's going to match up. And so those bonds are going to form, and that's all controlled by an enzyme.
Known as recombinant DNA.
So when you have the mixture of both DNA information together, it's a hybrid DNA referred to as a recombined or recombinant DNA.
Recombinant DNA plasmids are introduced into bacterial cells. Once they are in the host cells, the plasmids are able to replicate. Eventually, the host cell divides. Some cells may have recombinant DNA and some may not. A test tube containing recombinant DNA in host cells may be grown on agar plates. The bacteria may be plated on a selective medium containing the antibiotic ampicillin. The host cells undergo cell division and form bacterial colonies. Transformed bacterial cells are resistant to ampicillin and show growth. Untransformed bacterial cells die because they are sensitive to ampicillin.
All right, so in this example, it was showing bacteria that might be resistant to ampicillin. And you could see the ones that had the modified gene, they would live, while the ones without would die out. What are some advantages and disadvantages of this technique? So using genetic engineering, we're able to improve like our food quality, nutritional food quality. We can make plants that are resistant or weeds that can be resistant to pests or crops that can be resistant to weeds growing in it. It can improve the survival during like stress environment if it's a drought, area that deals with a lot of drought or too much rain. You could modify plants to be able to survive in those environments. You're able to create new materials for medicine or even for industries. The disadvantages, we're also seeing that insects or even bacteria are becoming resistant to the things that we've created. And so then we have a problem in that, as in how do we get rid of them as a pest? We are seeing that allergies could also become a problem. The materials we're inserting, can they affect someone or help create allergies in someone? It can also lead to new diseases that we didn't have before, this new combination. And so GMOs, genetically modified organisms now. So these are living things that have been modified. So that means that they have genes in them from a different organism. Advantages in that is that we're able to study certain diseases because of it. We're able to create vaccines in a safer way or test vaccines safely. We can aid in the production of some hormones, as I discussed before. It's boosting the pharmaceutical industry. We could increase like the production of lactose and cholesterol. We can increase this because we have organisms that are able to produce these things. The downside of it is that products in certain medicines are rare and they could also be expensive to do this. And there's little scientific evidence as to how effective some of this will be, the long term of this. Other things could be, is it ethical? People may question what rights do we have to genetically alter an organism, and what gets altered, and who gives the okay for that to be altered. And also we're seeing like new form of terrorism can be formed using, we have biochemicals that can be created using some of these techniques. All right, and so here's a practice question. I'll leave it on the slide. If we have some time, I'll come back and look at these because I wanted to get over here to cloning. And so cloning is when you're going to insert the DNA from one organism cell, but you're going to put it in an unfertilized egg. So in genetic engineering, we were taking a piece, a section of the DNA, and we're going to insert that into the new organism. So that's specifically the bacteria, to help that to make whatever the trait is we want it to make. But in cloning, we're actually removing the DNA and we're going to put it inside an unfertilized egg. So here we go. Look at the image. We have this organism, which would be our donor. So we're going to get the cell from the donor. And here we're going to get we're going to get an egg. We're going to remove that original DNA from the fertilized egg. We're going to insert now the DNA from that body cell into that fertilized egg. So here's the information that we want to clone. I'm going to use the red. And so now this cell that's fertilized is ready to go through cleavage. Remember, we discussed that earlier. Cleavage, blastula, gastrula to form the different parts of the body. And so here, the new fertilized egg has the genetic information from the donor. And so as this cell develops into an embryo, that embryo has all the new genetic information, which I'm going to try to represent my red dot. And as that organism develops, they will be genetically identical to the original organism. So your clone don't come out the same exact size. It has to go through the whole development process as that organism. So it's the donor cell will donate its DNA into the fertilized egg. The fertilized egg then goes through all the cell division process, a cleavage in the development of the embryo into adulthood. Here's another technique that we can use DNA for. We can actually use DNA fingerprint. Since DNA has, those are similar bases. We have adenine, thymine, guanine, cytosine is similar for everyone. We can cut the DNA at certain sections using an enzyme. Then we could compare that to other organisms. So we refer to that as a DNA fingerprint. We all have a DNA fingerprint. And so we could see where we could compare different organisms to the DNA we're trying to identify. So you could see this type of technique is used in crime scene. If you're trying to figure out who committed the crime and there was DNA left behind at the scene, you could compare that to a set of individuals. This is used during like paternity tests. So anything to identify someone based on their genetic information, you could use DNA fingerprinting. The technique of creating this DNA fingerprint is referred to as gel electrophoresis, where the DNA is taken through a process to where it's cut, and then using some sort of radiation and there'll be like a nylon fiber in this process. It's going to dye each DNA strand at a certain section, and you could compare whichever organism have the most bands in common with the original strand has more in common with the original strand. So in this particular example, we see that this person C, has more in common with the DNA sample that was found at the crime scene. So guess what? Individual C, you did it. And so once again, paternity, you have the female, and here's the child. And so you could compare parent 1 and dad 1 and 2 to see who's the father of the child. So it would be the one that has most in common. So he's got that in common, that in common. That's it. This one has none, has 1 in common. So this guy's 2, this one's got 1. So the most in common, that means father 1 is the father. It's going to be the organism that has most in common. So this is a simple form. It's more complex than this, but this is to give you an idea. Right? I wanted to do those practice questions with you. So let's see. Let's see here. Oops. Yeah, let's spend our last couple minutes doing some practice questions. And so it says practice question here. The gene from a human hormone is inserted into a bacterial DNA. So here's the recombined DNA that's then inserted back into the bacteria. It goes through cell division. So the process represented in the diagram, what's the name of this process? That's what's being asked. And that was genetic engineering. The original gene from the production of the human hormone was most likely removed from, where did it come from? It came from the human, the donor, for this genetic information. That one was so simple that it had me thinking twice. But yeah, it came from the human. State one possible reason why a gene from the production of a human hormone would be most likely placed in a bacterial DNA. Why would you put it inside of a bacteria? Think about that. Why in a bacteria? It's because bacteria is a simple organism. They reproduce quickly, so you're able to get the product quicker than if it was another organism. Similar to the to Mendel using the pea plant, because you could see the next generation quickly. Bacteria, they're simple to use, they're not expensive. Replication occurs a lot quicker than even the pea plant, and you could see the results right away. Here's another one. One variety of wheat is resistant to diseases. Another variety contains more nutrients of benefits to humans. Explain how a new variety of wheat with disease resistance and high nutrient value were developed. So what technique can you use if I want to make a wheat that is resistant and of high nutrient value? So I could genetically engineer this where I'll put the gene for resistance into the plant that's already providing the nutritional benefits. And so by putting this technique in, by putting this gene in, I will then produce a plant that's carrying both traits. I might have answered all 3 questions at once. Let me see. Describe how this technique will be carried out. I think I did. So removing that trait and placing it in. How about the third one? Did I answer that? Describe one specific difference other than stating that it does not work always. So one difference in developing a new variation using difficulty, not difference. So a difficulty could be that you could create something that would become a problem and we can't get rid of it. Or it may not work. Oh, it says besides that. All right, so something that we can't get rid of. It may not yield the way we want it to yield. It could be something that can have a negative effect on the environment. So all of these are possibility. And this is why genetic engineering, we're still working on it in the society and we're still making great headways with it. See if we have time for another question. And it looks like we do not. All right, so these are still their practice question. If you would like to go on your own and practice these. please do so. And I want to remind you that I am here this week again on Tuesday and Wednesday. I'll be here to review some questions. So hope to see you guys there. Hopefully you could join me. And I'll see you next time. Bye-bye.